I sincerely thank Professor Srivastava for the invitation, as well as Dr. Charni Pathak, my student, for the invitation to come and talk to you. I'm going to talk about a very special project of ours, particularly in biomedical application. This project has been running in our laboratory for 10 to 15 years. It's a long story. And this project also is something which is very close to our heart. I'm going to talk about designing novel metallopharmaceutical for chemotherapeutic application. We are looking out for some new motif, new drug, and that drug should have applications for chemotherapy or cancer related issues. When somebody wants to make a new drug, there are a lot of issues governing them. For example, these compound has to be bioactive. Bioactive to the sense that it should be acceptable to all, or it should beat all other existing medicines which are out there. It should perform well not only in vitro conditions but also in vivo, then on animal models. Then it should also have a lot of toxicological issues associated with the compound. It should be benign to those, and after a lot of screenings, then molecule come up to be labeled as drugs. This process is really very painstaking and long. For example, the number of drugs that come out in the market are very few and far between, whereas the number of compounds which are synthesized every year will be in thousands, hundreds of thousands, or in millions. But only few of them, of these compounds who are synthesized, only few will make it to the drug. And the process is very laborious, rigorous, time consuming, expensive. On an average, to give you a feel of a new drug that come out in the market, the company who develops it would have easily spent about $1 billion or even more. So a lot of effort goes in bringing out molecule which will eventually become drug. Now, given that challenge so high and so expensive, so time consuming, the major roadblock is nobody wants to work on the drug discovery track. They would rather take a molecule which is already approved by FDA, Federal Drug Agency, which is a body which approves the drug, and it's a body in the US. So they would take a biomolecule which is already approved by FDA. That means that all the toxicological parameters and criteria has been taken care of for that particular molecule, and then take that molecule and use it for some other medicinal treatment. So once that treatment gets approved, then there is nothing to worry about the approval of the drug because the molecule has been approved for particular medicine earlier on. This has been the main focus for many of the pharmaceuticals, even Indian pharmaceuticals. They would rather repurpose a drug. That is this whole feature of taking a known molecule which is already a drug and use it for some other purpose. There is a term for it. They call it repurposing a drug. So they would rather repurpose a drug where the hurdles are less than to take on an initiative where you come up with a new drug. And for many academicians also, this challenge is on the same front, that it is so huge a challenge, so long a challenge, that people may think that, okay, it's not even worth undertaking. Let's work on something which is like you can see the returns right away, not so uncertain as this would be. So while we were embarking on it, we thought that let us look at the same thing in a different way. We thought that we have nothing to lose, and there are not many people who really embark on new drug discovery track. To begin with, let me just acknowledge the people who had really carried out the work. I had this wonderful opportunity to work with some very young, talented, and meticulous researchers. The cancer work primarily is done by four graduate students during different span of their career. They were directly or indirectly related to this cancer project that I'm going to talk about today. I'm also thankful to all the funding agencies, primarily all the government funding agencies like DST, CSR, BRNS, as well as Indo-French collaboration, and then some private industries like Reliance, as well as IIT Bombay for all the funds, and also my collaborators. We work on a particular molecule which is shown in the middle, and this molecule is called carbene, and these are singlet carbene. And what is that supposed to mean is that they have a lone pair of electrons, and this lone pair they use to coordinate to metal center. One aspect of our work is on looking into the chemical reactivity or catalytic applications of the metal complexes of this N-heterocyclic carbene. And here we have our whole research focusing on the most unstable of molecule, and that too of its most unstable state, the singlet state, and try to make metal complex of that, and then look for the utility of the transition metal complexes of this carbene on two fronts. One is catalytic applications, which are in chemical catalysis. The other is biomedical applications, which are for developing them as metallopharmaceuticals in a new drug discovery track. So these have been one of the primary focus areas of our research over the last 15 to 20 years that I have been at IIT Bombay. So today's talk would primarily focus on the right-hand route, where we will look at the utility of these transition metal carbene complexes in biomedical applications, and particularly in cancer research, what's called translation. What does this mean? It means that if you develop some new knowledge in your laboratory, that has to go beyond the boundary of your laboratory. So the process by which the knowledge gets transferred from laboratory to the larger platform of greater utility, that process is called or is referred to by this word translation. But one thing to remember about translation is that one has to connect the small picture to the big picture. The drug that we are pursuing is a palladium complex of N-heterocyclic carbene. We have come up with a name for our compound for cancer therapy. It's called Palladacarbene, and the name sort of gives some sense of what is inside our molecule. Pallada means that it has to be a palladium compound, and carbene means that it is this unstable intermediate that I was talking about. The theme of the project that we are trying to develop over here is palladacarbene as a metallopharmaceutical for chemotherapeutic applications. How many new drugs are approved by FDA? For example, last year in 2019, only 33 new molecules have been approved under new molecule entity. These 33 molecules which were approved in 2019 may have started their journey about 10 or 15 years earlier on. What we will see is that of these 33, several of them are for cancer. Right now, the cancer is among the largest therapy area. Usually, neurological disorder was very common therapy area, but now cancer has overtaken them, and this has primarily because the people are living longer and older, and as a result tend to have more cancer. The problem is that the quality of life has been affected, in the sense that even though people may be living longer, but they may be having cancer, or they maybe have to fight cancer while they live longer. So that's also brings down the quality of the life they're having, even though they're better longevity and lifespan because of better medical system. All of these has contributed to cancer being among the largest therapy area. Cancer is indeed the biggest medical problem that one can desire to solve, and in that way, like it's very intimidating and very challenging. Under new molecular entity, there were 33 molecules. So what we see that of the 33 new molecules that were approved by FDA in 2019, about five of them are for cancer at different stages of cancer. Cancer is like a monster, a multiply-headed monster. There is no one type of disease. There are about 200 variants of cancer. 200 variants. So that 200 different types of cancer may occur. But if you look at all the cancer and the probability of that, you would see that only four or five types of cancer are very common. They account for 90% of other cancers. And remaining variants, they will occupy a small fraction of overall total cancer. So these cancers which are predominant, they are usually lung cancer, breast cancer, colorectal cancer, and stomach cancer. These are really the deadly cancers. And as you can see, this bar diagram, the blue one is the number of deaths. So let's say if I look at the lung cancer, it tells me about 1.2 million people die of lung cancer, and then the red bar corresponding about 10,000 people, they do not die, but they are called incidents. So you can see that lung cancer is quite deadly. Breast cancer also, you see the blue line is not as big as the red line. Red line means they are affected patients. Blue line means that they have perished in cancer. Colorectal cancer, you see the blue line is longer than the corresponding red line. Stomach cancer also, you will see the blue line is larger than the red line. So that means in all of these cases, the number of people dying would be more than number of people living with the cancer. So these are really lethal cancer. And if you look at it, the liver cancer, the red line is like a dot. So that means that if somebody has a liver cancer, most likelihood the person would die. So look at this. The pancreatic cancer is over here. So the red dot is almost nothing. So this is the lethalest of all. Like if somebody has a pancreatic cancer, most likely the person will die. The lung cancer also we see that very little people live. It's not a blue means all death. Same with stomach, colorectal. In that way, breast cancer, many people survive. These five cancer will account for 50% of the total cancers and the total deaths as well. So these are probably the most important forms of cancer that are seen today. So in addition to what I just mentioned above, these are all solid cancer. Solid cancers means that you have a solid mass, a solid tumor somewhere. It can happen anywhere in your body, inside your body, liver, lungs, colorectal, anywhere, but it's a solid cancer. Another type of cancer is called hematological cancer. So these are actually blood cancer. Like, it's not a solid, it's just the cancer is blood and also contribute to significant clinical problems. Across the globe, every year, about 12 to 14 million people die. And cancer deaths accounts for 13% of the deaths worldwide annually. So that's really a very huge number. Uh, it's known as a silent killer. Medically also silent killer in the sense that when it spreads, the symptoms are very mild or subtle to the extent that they are not noticed at all. They are so subtle until when it becomes the last point. When they are detected, it may be in so advanced stage that it is very difficult to save the patient. So it is a silent killer. And the many-headed monster, 200 variants. It is a faceless enemy. With cancer, everything is so uncertain and also unpredictable. There is no real answer to cancer. And also, the cancer is gaining more prominence. That's because increased longevity leading to aging of population. There has been a substantial medical success that people are living longer so that we are preventing many of the disease. So the mortality rate is increasing, but this increased longevity has also brought in people getting afflicted with cancer. Now, there has also been a change of thinking. Initially, the approach was that if you have a cancer, we'll have to find medical treatment completely wipe away the cancer. It seems that in certain cases that it becomes impossible to wipe away the cancer unless the patient dies. It is very difficult to wipe away the cancer in many cases. So there has been a shift in medical thinking. The shift has been that make the cancer slow. It will be something that one lives with it. It becomes a chronic disease. There has been one line of medical approach where they are trying to focus on changing cancer from a lethal disease to a chronic disease. Now, in that line of thinking, that since the cancer has changed to a chronic disease, then there will be a lot of people with cancer. So that also leads to spread of the cancer because it does not eliminate cancer. Because of all of medical treatment as well as longevity, all of this is leading to spread of cancer. Here is a graph. There are four lines: red, blue, green, pink, and this represents the stages of cancer. So that means that if you have breast cancer, yeah, which is blue, blue means stage 0. The cancer is just started to develop, but not spread. So then stage 1 and 2 is where the cancer is limited to a localized cancer. Stage 1 and 2 is called localized cancer. That means the cancer is limited to one organ, but it has not spread. Then comes stage 3, yeah, which is called regional cancer. That means the cancer has spread from the primary organ to other nearby organs. And then comes stage 4, which is shown in green, that is called the distant cancer, which is the cancer has spread. And then comes stage 5, which is the cancer, like is all over. So there is not much information. So cancer, depending on what stage it is and what type of cancer, that will determine the survivability. For example, early detection, if something is detected at stage 0 or stage 1 and stage 2 where cancer is still localized, then it is easier to treat. So what this graph shows, that if you have breast cancer even to stage 3, which is regional, you will survive. This white line is the survival. But let's say if you have pancreatic cancer, which I just said, the most deadliest one, even in stage 0 or 1, you may not be able to survive. Similarly, prostate, you can survive up to the pink, which is the on stage. So depends on what type of cancer one has, how deadly the cancer is, and what are the 4 stages. First is stage 0, which is just initial in situ cancer, then localized cancer, regional cancer, distant cancer. So if one has to target a medicine, they have to target below the survival region, somewhere over here, for patient to survive. What are the combat strategies? Actually, there are several. For example, right now, this detection and diagnostics. So what they do, no, they do imaging. They go and take image of the body part and then match it with artificial intelligence with the cancer image, and then decide whether the person has a cancer or not. Then comes surgery. If the cancer has not spread, if it's still in stage 1 and stage 2, then you can cut out that organ and still the patient have a good life. Or it can be drug discovery, also chemotherapy, which is surgery less way of targeting cancer. So we thought of focus our research on this chemotherapy bit. One thing we know for certain about cancer, even so much of unpredictability, uncertainty, but among that, one thing is for certain. What is that? There cannot be a single strategy or answer for cancer. So you cannot develop a pill, let's say aspirin or something, and then you say take the pill and then you will be totally cured of cancer. No, because cancer is of so many types, so there cannot be a single answer, a single medicine for all types of cancer. That much is certain. And another important thing is that no single expertise can tackle the menace of cancer. Cancer is so big that only chemists cannot do, only biologists cannot do, only computer experts cannot do. So it has to be a multidisciplinary approach, that you have to cut across several disciplines, all targeted towards cancer. So everybody has to chip in. People who cannot understand each other's languages, they are in totally different area. They also have to, doctors, biologists, clinicians, medical experts, toxicologists, pharmacologists, chemists, synthesis chemists, everybody. Everybody has to come in and then develop the medicine. So it can, this much is certain, absolutely certain, that no single field can come up with a cancer. So call of the day, what is the need of the hour to call for a transdisciplinary combat strategy, that it has to be across the discipline. Many discipline has to, so whatever you want to do, it has to be done together. For example, then what is the big questions? Can we make complementary expertise to work on one cause? There's so many different people, different, can they work on same cause? And can we make so many different experts to be excited about one work? There has been instances where people have gone up to the challenges, risen up to the challenges. People of different expertise come together and try to solve these problems. And cancer is one such that remains to be done in that way. So right now, there is we don't have the liberty or choice, but that is the only way forward, that we should work together. What is chemotherapy? Chemotherapy is last line of defense. That means when the disease has advanced to the patient, at that stage when all other options have failed, so then chemotherapy is brought in. Now there is a reason for that. Why chemotherapy is not given earlier on? That is because first, earlier on, the cancer is not developed enough to understand that it is cancer. And also, chemotherapy means what? Giving chemical compound. Any artificial compound is toxic to the body. So chemotherapy will have a compromise on the quality of life. So there will be hair loss. There will be so many bad things which will result from chemotherapy. So chemotherapy is not brought in the first line of defense. Then they try all other techniques. When all the techniques fail, then chemotherapy comes in. Now that also is difficult on chemotherapy because by the time when chemotherapy is called, that time the maximum damage has been done. The patient has reached stage 3 or stage 4 or even stage 5. So at that point, like it has to be a really a great effort for chemotherapy to salvage the patient. Nowadays, they also come up with a new thing called combination therapy, and they try combination of two, three drugs instead of one drug. Try to affect it. This also has advanced a lot of combination therapy. Once you give chemotherapy, there will be a lot of side effects. The doctor who gives chemotherapeutic agents, he does makes a prescription thinking about what are the side effects the patient may suffer, how the quality of life will be compromised, and do that. And even for chemotherapy, the price cancer patient pay for employing this aggressive technique is very high. That means that even though he may get rid of cancer, but he has to endure a lot of other things like hair loss or other organs may get affected or damaged. So the quality of life really goes down, and that's why the chemotherapy, even though is a solution, is not really a great solution. So cumulative toxicology burden of chemotherapy resulted in dry treatment to spare healthy cells from bystander damage. So now there is a chemotherapy which is targeted chemotherapy. That means that there is an effort to develop chemotherapeutic agents which will only kill the cancer cell and will have less effect on the living cell. So now, if you want to find a solution, you need to understand the question first. So, what is cancer? So, one want to understand cancer, you need to know what cancer is to find a solution. Now, cancer is a characterized autonomous cell growth, invasion, colonization, neogenesis, reduced cell death. So what does cancer mean? That the cancer cell divides at its own rate. It is like autonomous. It doesn't feel to be associated with the part of the body. It just become like self-replicating, self-dividing system, and it sort of rebels from the body. So the body grows in one way, the cell grows at a faster rate, and that is associated with three parameters. So for some reason, the cancer cell becomes self-sufficiency in growth signal. So when a cell divides, there are a lot of biological pathways that occur within the body across the organ. But for some reason, cancer does not acquire all these signals in a cell-sufficient manner. It does not require to be in tune with the rest of the body, and that's why you result in formation of huge tumors. Then insensitive to anti-growth signals. Now, body also have a lot of biochemical pathway which will restrict the growth. But for cancer, they don't obey these anti-growth signals. They become insensitive. Then cancer can take over tissue invasion and metastasis. So cancer can spread, can affect neighboring tissue and then spread further. So that is a property. Slowly it starts from something and then keeps on expanding. Limitless replicative potential. That means that unlimited time it will multiply much faster than what the body requires. Sustained angiogenesis. Now, when it multiplies, it needs a lot of energy, and to keep the you need new blood vessels and all of this process it can does it by itself. And also, body has a mechanism in which it kills the old cells, and this mechanism called apoptosis. So, cancer cell can avoid apoptosis or evade apoptosis. In this way, when the body is killing the cell, other cells are being died, but cancer cell body cannot kill, and the cell grows. So, this is what cancer is. That's like uncontrollable colonization. All these words that is mentioned over here that defines cancer's properties. Now, as I said, that there are two types of drug therapy. One is the chemotherapy where you try to kill the cancer cell, and now there is a new kind of therapy which is called targeted therapy, which focuses on all the signals, as I mentioned, that defines the cancer cell. There are some proteins and some biochemical signal pathways. So these medicines target that so that the cancer stops dividing, or if not starts slow division, and as a result becomes transferred to a chronic disease. And this is what results in translation medicine. So they, instead of like completely eliminating the tumor, they try to freeze the tumor, that the tumor does not grow. whereas it will just suppress the growth, whereas chemotherapy will target towards shrinkage of the tumor, completely eliminate the tumor. So if you just freeze the tumor, then you are changing cancer from a lethal disease to a chronic disease. What we thought is there are a lot of organic drugs for cancer, for example, Taxol, and then we have the inorganic counterpart. And what we saw that the organic ones are very potent. That means in nanomolar range. And this inorganic one, cisplatin, which is a famous drug, that is in micromolar range. So our idea was why the inorganic concentration is more, which will kill the cancer. And we know the answer because for organic drugs, it works on microtubules, which are present in cytoplasm. So the drug has to just penetrate the cell wall and go to cytoplasm. Whereas for the inorganic one, it works in nuclear. So they have to not only penetrate the cell wall, but penetrate the nuclear membrane as well. So more concentration is required for this. And then the question that we asked on the project was, can we make inorganic drug as active as the organic drug? So can we use palladium? So instead of platinum, what is the scenario beyond platinum? Since this cisplatin affects binds to DNA and DNA is chiral, so we wanted to know that what if we make some chiral drug? Will we have any chiral induction in that? Now, once you make a drug, then there are ways in which you want to deliver the drug. So there are various strategies. For example, improve the aqueous solubility, or to make it more effective, you do a controlled sustained release that slowly release the drug. Then circulation in human body, you put it in the blood, or you just inject tumor specific delivery, just inject on the tumor, and then the long term efficacy of the cell now metal-based complexes. So what we thought that we want to make focus on the first idea, simple chemist perspective, to improve the solubility of the drug that we want to synthesize. So right now, if one looks at the drug scenario for cancer, it is only inorganic ones are only platinum. So various forms of cisplatins are known. For example, cisplatin, carboplatin, metaplatin, streptoplatin, so many. So four. So the only player in the market is platinum. And also this particular tetraplatin, this is only platinum 4, Westerol platinum 2 and Westerol platinum 2, and this is given orally. That means the others are given through blood injection, intravenous, very painful, and this one is given orally. So that's sort of an advantage. And it also shows that platinum 4 can be used for making the drug. Now, how does cisplatin work? So cisplatin binds to DNA. Cisplatin also triggers apoptosis. As I said, that apoptosis is a mechanism, normal mechanism of body of killing the cells without a history. When the dead cells are removed, new cells will be formed, and there will not be any history of the dead cell remaining. The body replaces the dead cell by living cell, and in a natural way, so that people don't see the dead cell. So that kind of apoptosis is good, natural way of healing. But if the dead cell remains and they cannot be removed from the body, then it becomes tumorous, and that is called necrosis. If you want to design a drug, so the drug has to bind to DNA and also has to trigger apoptosis. So limitations of cisplatin, like there is reason that people should go beyond cisplatin. The number of tumor which is effective for cisplatin is less. Solubility is extremely bad, 1 milligram per microliter. So higher concentration is required. It has side effects like vomiting and hematological toxicity, and also drug resistance. Cancer becomes resistant to cisplatin treatment with time. So there are a lot of drawbacks to cisplatin. Our motivation is that since non-platinum drugs are not known, so we have this whole periodic table. We are a bunch of synthetic chemists. We love to make molecules, so we have a whole periodic table to play with. Of the various elements, right now, ruthenium, titanium, gallium have entered the clinical phase, and iron, cobalt, gold are in the preclinical study. So these are potential candidates. And what we thought that we will target palladium. And here I would like to show that we have the same two molecules but a different cations. One is organic cation, another is inorganic cation. And since this compound with this inorganic cation is more soluble, this is 35-fold more effective than the organic one. So we thought that we are in the right track, but if we can make more soluble inorganic palladacarbene complexes, that should be extremely good thing. And there has been for photothermic therapy the use of palladium complexes. So what we want to focus our molecule in a new drug discovery stream is this palladium. And palladium, there are some reports that it has cytotoxic properties, and also palladium is genetically labile, and it can stabilize various kind of charged tissues so that it can interact with DNA and should be a good candidate. Also, palladium falls below platinum, so would be an ideal candidate to do. These are the reasons I've already said. I'm not going to say why we chose Metallocarbene. They are strong sigma donors, stable complexes, easy to find. So we thought that we should use carbene complexes of palladium, which we have given a name as palladacarbene for this anti-cancer studies. Now, as I said, that cisplatin was known and transpyridine was a compound which was in the clinical studies. So we thought that if we keep on replacing pyridine with carbene, first one carbene, then two carbene, this should be extremely active for cancer treatment. And we synthesized all of these complexes. I'm going to skip that. And what we saw that indeed, when they synthesized these biscarbene versus platin, we saw that our biscarbene is about 2 to 20 times more active than this cisplatin. So this is 8 micromolar, this is 4, so it is half. This is zero point 0.8 micromolar, 16. This is 20 times more active. The lower the concentration, more the activity. And then this one is 15 to 1. So that shows that this carbene is more active with two carbene than the cisplatin. This is more active than the cisplatin. So that's good. And then after our report, there were some other reports of different carbenes which came up, and they also found activity similar to us. And there also all of these activities were in micromolar range. Now, as I said that how does our complex work? We have done several experiments. This is a cell cycle, like starts with G1 phase, cell grows, then synthesis, replication of DNA, then G2 cells prepares to divide, and then mitosis M. So through various immunofluorescence studies, what we have seen that our complex sort of stops at G2M phase. So that means that it does not, it freezes the cell in uh, the gap to mitosis. So it stops the cell at the just at the G2M phase before mitosis, and hence it affects the cancer. So that is a very nice thing to know that how does this compound work. This is a general cartoon which shows how many types of different chemotherapeutic pathways are known to kill cancer. The first one is platinum complexes, which sort of goes to the first breaks the cell wall, goes to the cytoplasm, and then enters the breaks the cell membrane, nuclear membrane, and goes to the nucleus, and then binds to DNA. And after binding to DNA, the DNA transcription stops, and then apoptosis or the cell death starts. So this is the pathway that our compound is showing. Now there is another parallel pathway which is also effective, and these are mainly seen for gold kind of metallopharmaceuticals. So the gold chemicals, instead of going to the DNA, what they do is they go to and attack mitochondria. So these cancer cells divide faster. Cancer cell requires a lot of energy, and this energy is supplied by mitochondria. This gold drug, what it does, it destroys the mitochondria, and as a result, the energy which the cancer cell requires to divide is lost, and then the cell cannot divide further. So this is the second pathway: mitochondrial disruption, cell membrane rupture, react oxygen species, and then program candidate. So there are two ways by which the metal pharmaceutical works. One is by going to DNA and stopping the transcription, in which by our compound works. The other is destroying the mitochondria, which provides energy for the division. Now, what we thought is as the DNA is chiral, so if we make this our complex chiral, then we should be able to see a variation, like one particular enantiomer being more active than the other. So with that in mind, what we did is we went back to the drawing board and tried to synthesize molecules, but this time we want to synthesize the molecules with carbenes, which will be more soluble, generate lead, and also will probe the chiral nature of the interaction. With that, we sort of made 5 NMPO pairs. There are actually 10 compounds, and then two are bromide and two are with polar moiety trifluoroacetate. So this was put in place deliberately to ensure that the compound become more soluble, and these are NMPO pairs, and then we put some chiral methyl group. So the idea being that let us see if we can see any chiral induction, chiral recognition as to one NMPO bar being more active than the other, and went and prepared all these 10 compounds, both the enantiomers, the plus and the minors, and got them structurally characterized. And then finally, we tested them. Now, when we tested them, so red is for one and green is for another, what we saw that we did not see any chiral differentiation. both the enantiomers were equally active. There is a reason for that. I think we knew at this point, but at that point we were a little bit perplexed as to why there is no enantiomeric differentiation when this is binding to DNA. But what is more important is that we came up with a trifluoroacetate molecule which had now extremely active. Both the enantiomers are extremely active now in nanomolar range. So we have broken that barrier of micromolar for inorganic cisplatin and everything with our palladium system, with our carbene. Now we can go to hundreds of nanomolar. So this itself is a significant discovery, and we saw the green one is more active than the red one. So we focused on our study with the green one now. So what we did is we took this further and then studied these two compounds across cisplatin. So the big one, what we saw that these are the cisplatin data, and these are small are the data for our both the enantiomers, and we see that these compounds always hit cisplatin twice over, both for cervical cancer as well as for breast cancer. Our compounds have much lower value than that of the cisplatin. We then looked at the scope of applications across various different cell lines. We saw that has extremely good activity for breast cancer, cervical cancer. These are like main, very important cancer cells. As I said in the beginning, that they account together, they account for 50% of all other cancer deaths, skin cancer, lung cancer. But what is most important is that it affected this memory multi-drug resistant cancer. So this cancer cell has now become resistant to other drugs by virtue of modification. Now this our drug is also effective on that. So that is sort of very good news. And then we also looked at the selectivity. And what we found that these are the non-cancerous breast cells and these are the breast cancer. So our is more effective on the cancerous cells and less poisonous on the less toxic on the normal cells. Same thing with the skin cancer, and this is the non-cancerous skin. So this is more effective on the skin cancer. So what we see in the end that this molecule that we have developed is about 16 times, 8 times more selectively toxic towards cancer cell than for the non-cancerous part. As for the question that why there was no differentiation in the biological activity when we went from one enantiomer to the other, and the answer to that is that this major group, the dimensions of the major group and the minor group is much bigger than the dimensions of the chiral moiety. There is a mismatch in the size, and hence the chiral recognition did not occur as planned. And there is another possible explanation is that the compound may decompose as well. So that also at this point I cannot rule out. One good thing is that we have been granted a US patent. Actually, we had applied for it in 2018, and in by 2019, within 1 year, few months, the US patent was granted. We can take it further because all intellectual property rights have been protected. Another important highlight of this story is that this is totally IITB endeavor, everything in IIT Bombay, and in that way it is a complete made in India initiative. And as I said that given the first drug discovery challenges are so few and far and so demanding that there is not many attempts even being made because of the dauntingness of the task, and we consider ourselves extremely fortunate to broken the first barrier, and we are actively pursuing it at the moment with different clinical conclusions. With that, let me just summarize. We have developed a completely new molecule from IIT Bombay, fully new drug discovery track, that too for cancer, fully in-house endeavor at IIT Bombay, and this also is intellectually protected, completely nascent, original, secret platform for further development in a new drug discovery track. What we have done, we have gotten hold of a new motif. We promoted a new metal. This has a broad range of activity, several cancer cells. Selectivity towards cancer cell does not, it affects normal cell less, but it kills cancer cell more. And also, we have broken the barrier of micromolar and gone to a regime of submicromolar activity. A lot of work remains to be done, for example, in vivo, in the ex vivo validation studies on animal models, then the formulation and the dosage studies that are taking some academic studies like computational docking, DNA binding, those are to be done. Also, further optimization of the models to be undertaken, that needs also to be done. And said that we have given a name, it is called Palladacarbene. And where Palladacarbene, which space does it want to fulfill? High efficacy at low dosage, low toxicity, effectiveness against wide range of cancer, active against multi-drug resistant cells. With that, I come to an end of this lecture. I would be happy to take any questions.
Nancy Jain has asked, is chemotherapy approach selective?
There is some degree of selectivity. Chemotherapy is not 100% selective towards cancer cell. No, the gradient of selectivity depends on different cancers as well as depends on different medicine. So some degree of selectivity, some means how much, maybe 20%, 30% more, or 70% more, but it's not like 100%, 0%. No. So that is sort of the limitation of chemotherapy. A chemotherapy, since it is not completely selective, so there is always a side effect to chemotherapy on the patient, and that brings down the quality of life of the patient. We will have a lot of things, hair loss, chemical imbalance. Sometimes they get blood cancer also. So there are a lot of very terrible side effects that chemotherapy brings on to a patient. That is why even though the longevity may be prolonged, but how you live, that also is important. You don't want to live in a very poor condition. So you want to increase the lifespan of a person, but if a person lives in a miserable condition, then is that acceptable? Or to what extent would you allow the person's condition to continue? So there are some ethical questions also, moral questions also. Those are there apart from the medical challenges. Partially selective, there is combination therapy. So right now, as I said, no, there is a alternative approach. The alternative approach is not try to shrink or kill the tumor, but try to freeze the tumor. When you freeze the tumor means that you stop its growth. If you stop its growth, no, it's sort of an extra appendage that you carry. But if you can just stop its growth, you don't have to really fully kill it. Then it becomes a chronic disease. It's forever. Chronic disease. It's there. It's not eliminated, but they're living with it.
Thank you, sir. There is another question from the same person. She wants to know how long does it take to recover from PDT?
I'm not a medical expert, so depends on what extent, at what stage the cancer is when you do PDT, and how successful is your PDT. If the cancer is localized, like in stage one, stage two, like that, and with PDT, like you completely eliminate, then the relapse may take long time, 5 years, 10 years, like that. But if the cancer is in stage four or stage three or stage four, when and there you do PDT, then it might not have any effect. The mortality of cancer depends on two things. If somebody has cancer, no, don't get unnerved or just because you heard the word cancer. You should know what type of cancer, because success of living, chances of living depends on cancer type. First thing is what type of cancer. Second thing is what stage of cancer. If it is not so lethal cancer, then a person can live even in stage four of that cancer. And if it is one of those rare but very lethal cancer, even stage zero or stage one will kill the person. So it's very cancer subjective and stage subjective. At advanced stage, the only solution is if you want to kill the cancer, the patient also dies. Like it's very painful.
There is another question from Marina Das. She wants to know, can differences in cancer cell metabolism be exploited? I hope from curing cancer she wants to mean.
One approach is to target those signaling pathways to stop those. Now, if you stop the signaling pathways, you are affecting the rate in which the cancer grows, and try to slow the growth and convert to a chronic disease. Now, in that can be say like seen as one way of manipulating the natural growth of the cancer. If you are not targeting that signaling pathway, cancer would go by its in its own way. But now that you are targeting treating with the signaling pathway which affects cancer growth, you are somehow manipulating the cancer growth. And try not to grow it bigger. You just have to arrest or freeze the tumor. So in that way, there is targeted therapy.
Thank you, sir. Thank you. Okay, so one question there from Rajendra Kumar Dikshit. If somebody facing brain cancer and radiotherapy has not showing enough effective, then what are the options? It's more of I'm sorry, it's more of a medical question, but I hope you can put some light on that.
there are two ways. One is that you kill the cell in situ, kill the cell within the system, or you perform surgery. Okay. Now all these things, PDT, radiotherapy, they are using light. Chemotherapy is chemical. So you either use light or radioisotopes or toxic chemicals to kill the cancer within the body. Okay, and let then the body deal with it. Uh, Either you use light or radioactivity or toxic chemical. Okay, and give it to the body, living body, and then kill the cell and let all the toxin everything be removed and let the body handle it. But the cancer remains in the body. Okay. So if radiotherapy fails, people, I don't know if there is a chemotherapy available for brain or not. I don't know. Uh, and then when all of those doesn't work, then and then the alternate option, what option? Surgery. Surgery is only is not something which is good. No, you're going to cut something out. So surgery is only successful if it is localized. If the cancer has spread, then you have to cut the whole body out. Then the patient will die. So if it is a localized cancer, localized means that it is just restricted, has not spread, then surgery may be though painful, but maybe only like completely 100% cure of maybe. But brain is just something. No, brain surgery also is one of the most difficult of all surgeries. I mean, as per your question wise, that it may be surgery provided that it is localized. Any surgery is feasible on cancer if it's stage one, up to stage one, stage two. Beyond that, surgery will not be applicable. So that's why early detection is very important. Early detection, early through imaging or something. If it's detected early, then it is better. Then surgery will be possible.
Thank
Okay.
you, sir. Thank you. Is there any other questions?
Hello, sir.
Hello.
Thank you for your informative session, sir. My question is why platinum is the most used inorganic element in the anti-cancer drug?
Because it got developed. It had cisplatin, it got developed, and then all the research was funded that. So there was early breakthrough with cisplatin, and then a lot of went into cisplatin. Also, the advantage with platinum was that platinum is not extremely like not really toxic like the people like mercury or something that people will immediately die. So their body had some amount of tolerance level, and when the cisplatin was found to be effective through binding to DNA, then a lot of research went into make the modifications of cisplatin, carboplatin, bisplatin, that platin, and they also showed quite a bit of promise. So it's sort of like all the attention then just focused on that, and that kept on developing, and then it went on till now there is a need that cisplatin is seeing its limitations. Now that it has developed, it is saying that okay, it cannot go any further beyond. So now there people are sitting back and seeing what else can it be. Now, if you think of it very principle that what is the principle cisplatin is working on? It is some toxic chemical which goes to cancer and kills the cell. So would be any other inorganic metal complexes. They would all be toxic. If they go back and step back and do it, then it will also. But the point is whether it will get the same amount of effort, same amount of funding. Now it's something which comes up that has to outbid cisplatin to be able to consider. Yes, there is as I said, no, there were six metals. Three of them like gold, rhodium, ruthenium. They're in the preclinical study, gallium. So there are other metals which are at different stages like of FDA approval.
Thank you, sir. Sir, is there any other questions?
Sir, I
If
have one question. What is the best drug delivery system for cancer?
As I said, no, think for cancer, there cannot be a single answer. Like if you say best means one, but there cannot be one because there are so many types of cancer. So one delivery cannot be effective for all. Okay. But I can say which are the promising ones. Right now, they're trying to make drugs coated in various kind of nano formulation as a nanoparticle with some carrier which goes inside and then delivers. That some way is gaining a lot of research attention currently. So formulation, particularly using nanoparticle as a drug delivery vehicle, that is a sort of recent kind of craze. And also drug delivery is dependent on cancer. So if the cancer, let's say if it's the blood cancer, you need one strategy. If it is lung cancer, one strategy. If it's skin cancer, so depending on the localization, how to reach that, how to, oh, and in some it may be just you just inject directly into the cancer quickly. So that depending on cancer, depending on patient, there are various strategies. Sustained releases and in which you put it in the cell, but the compound does not immediately dissolve or go to affect, but slowly over time, little bit goes and kills the cell. Sustained release. That is also an interesting way in which they package the molecule in such a way that the drug is released slowly over time. And then the older method of intravenous injection in the blood, those are also there. So there cannot be, I think, singular answer. Another project we're working right now is on blood cancer that I did not talk about today. So there arsenic is used, arsenic oxide, dioxide, AS2O3 and all, and various formulations of those. So those people tell me that right now the drug delivery using nanoparticle is kind of uh, the most important way of drug delivery system that they feel. So my understanding is that may be the best one so far, but again, like depends on what type of cancer, what kind of disease, what stage on it.
Thank you, sir.
Is there any other questions? If not, sir, I have a question. So you talked about some some complexes where carbene was used as a ligand. So is there any other kind of ligand that's good for this kind of complexes?
For biological activity or what?
Uh,
Yeah, a lot of sister systems are there with lithium. They're non-target systems, you know. And then cisplatin, you see, you have two ammonia bind and two chloride. There's nothing carbene about it. So carboplatin, so all the variants, they are not carbene. So the many ligands are there. Then I showed trans bispyridine dichloride. There are only two pyridine bound to that.
Thank you, sir. Thank you. Is there any other questions? Okay, so if there is no other questions, I hand over the session to Dr. Chandni Pathak to give a vote of thanks to Professor Prasenjit Ghosh.
Thank you, everyone.